Good day. Welcome to the Waters and TA Instruments Medical Device Series. This series aims to show you how it can help you understand your medical devices better from a chemical and materials characterization perspective. This can then be applied to product development, to processing, to regulations, to troubleshooting. This is presented each week by an expert in their respective technology. For this webinar, we encourage questions, and we'll have these experts on hand to answer as many as we can in the time we have. Those we can't get to today, we will follow up with afterwards. Please post questions in the top left window and we'll answer at the end of the webinar. There are links to resources in the bottom window and a survey in the top right, which we would appreciate you completing. I would also invite you to register for future episodes of this series. This series will comprise of multiple sessions, one a week. These will cover various aspects of medical devices and the materials used for these devices. I will highlight how some of the technology from Waters and TA Instruments can answer some of the important questions that you have. We open this series with a focus on the chemical characterization and some of the regulatory changes that have happened of late. We then moved into understanding the mechanics and the strength of biomaterials and the tissue that will be replaced or that will surround a medical device. And after that, the fatigue and durability testing of medical devices. This week, we'll be speaking to the rheology of soft biomaterials. I'm Ben McGreath, responsible for the medical devices market at Waters. I'll be your host today and for each session of the series. Today's presenter will be Peter Harder. Peter is a senior application scientist for rheology with over 27 years of experience spanning both industry and here at TA Instruments. He spends his time engaging with the market, helping our existing customers, potential customers, and in general sharing his knowledge of rheology in his regions, which include Northern Europe. This series covers a variety of aspects of medical devices, from the final devices themselves to the components to the materials used. At each stage of development, there are key challenges and activities where chemical and material characterization is crucial. Your research or testing needs might be on the devices themselves, understanding the final attributes, the stability or the performing quality control, or you might be more interested in understanding the material that forms the device, how changes here impact the end performance and usage of the device. Through investigating the chemical and material nature, you will gain a much better understanding of your medical device. Today we'll hear how rheology can be applied. Over to you, Pete.
Welcome to this presentation on the rheology of soft biomaterials. In this presentation, I'd like to give you an introduction to rheology, to briefly discuss TA Instruments' rheometer range of instrumentation and test methodologies, and for the bulk of the presentation focus on rheological applications with biopolymers and biomedical materials, with examples of hydrogels and creams, adhesives, drugs, capsules, and biopolymers. So what is rheology? Well, we're all familiar with liquids, materials that are easy to pour or decant. At the other extreme we have solids, like bones, metals, or capsules. And in between we have what's called soft solids, things that range from creams, lotions to ointments and adhesives. Now, rheology is the science of deformation and flow of matter, and with these we can categorize materials as being on a liquid, viscous range to a solid, elastic range, and anything in between is viscoelastic. If we consider one of these rheological extremes, the viscous properties of a material, it's often represented by a dashpot. This is where we put a weight on top of a plunger. The plunger sinks at a constant rate based on the resistance of the liquid it passes through, the viscosity. When we take the weight off, you won't see the plunger float back to the surface, but it will stay where it finished. So if we represented this behavior graphically, when the weight was placed on the dashpot's plunger, we would see a linear movement with time. The strain, the distance travelled, would increase until the weight was taken off. At that point, the plunger would stay in a fixed position. This would have dissipated and wasted energy due to viscous flow. At the other extreme, we see an elastic behavior, represented by a spring. When we put the weight on the spring will stretch. However, if we take the weight off, the spring will recoil back to its original position, therefore storing energy. So if we represent this elastic behavior graphically, what we see is on the addition of the weight to the end of the spring, we see an instantaneous change in strain. which remains constant until the weight is removed and we return to our original position. This now gives us our two extremes, viscous and elastic behavior. However, most materials have a bit of each. They are viscoelastic and show both viscous and elastic properties, energy dissipation and energy storage. Rheology has many roles in real life, from processing and manufacture, where materials processing behavior can be monitored throughout to understand and optimize the process. This involves taking raw materials for QA, monitoring reactions by the intermediates, and finally evaluating the final product by QC. We also have formulation development, where in R&D we can actually make and monitor materials performance. By changing the formulation, we can make a better product. Some of these testings are related to some subjective tests, looking at the performance of the material itself, and rheology has been able to give us a better way of accurately grading the performance of materials. And finally, rheology can look at the structure, the makeup of a material. By modelling its material structure, we can look and understand things like molecular weight distribution, particle size distribution. So let's briefly look at TA Instruments rheometers and discuss the common rheological experimental methods. TA Instruments offer two types of rotational rheometers, the HR series 10, 20 and 30 and the AVES G2. The HR series is what we call a controlled stress single head rheometer, which means combined motor and transducer CNT technology, as shown in this graphical representation. The AVES G2 is unique in it's a controlled strain instrument, how far does it twist the sample, and is a dual head, a separate motor and transducer configuration, SMT. And as the graphic shows, this is a purer form of deformation, as the motor deforms the sample and we measure the sample's response in a separate transducer. Now when it comes to using these instruments, we have a range of geometries which configure the shape and form of the sample. They enable us to measure materials that are from water through to stainless steel, with concentric cylinders measuring very low to medium viscosity materials, things that are easy to pour and decant. Then these are best choices for unstable suspensions. The cones and the parallel plates are used for very low to high viscosity materials, though the cones are the best choice for colloidal dispersions and suspensions. The parallel plate has the added benefit of being able to work at different gaps, and as a result is the best choice for particulate materials, gels and soft solids. In terms of the torsion rectangular, the 3-point bend DMA and linear tension functions, then these are best suited for measuring solid materials, whether they're in a bar form or thin films. There are many different experimental modes available on these rheometers. With the unidirectional testing, we usually refer to flow. Here, we can ramp the stress or rate and monitor the viscosity change of the sample. We can also do this as a series of individual steps, and so we can see if there's any time dependency on this process. We can maintain a constant shear condition in peak hold or stress growth, and we can do exactly the same but change temperature in the background, and so we can look at mixing or processing conditions. There are some other unidirectional test methods available. The creep testing is where we can simulate processes like gravity acting on a material and measure the sample's deformation over time. Or we can deform the sample a set amount and monitor the stress as it relaxes away. And finally, we have the ability to measure the force generated when we separate the sample to pull apart or to penetrate the sample in axial testing. So when it comes to flow properties, we look at a rotational mode where we're monitoring the viscosity's relationship to the shear conditions the sample is exposed to, whether that's how hard we deform the material or how fast the rate we deform it. And it should really be of interest to anybody in moving a material from A to B. whether that's pumping it through a pipe or injecting through a needle, whether it's between two surfaces, like fingers applying a cream to your arm, or whether it's just storage conditions of a material, or for example, soaking into the skin post-application. A modern rheometer can measure a wide range of viscosity values, from air all the way through to solid materials like bitumen. Now, Pascal seconds is the usual SI units, but some may be familiar with millipascal seconds or centipoise, where there's a factor of 1,000. And it should be remembered that if you take a material and change the temperature, you'll often see an associated viscosity change. Where we increase the temperature, we see a viscosity drop. Having performed a flow experiment, we can plot the sample's viscosity with the shear conditions, and we can use this to predict the sample's behavior or performance during processing or other applications. The viscosity shown here is independent of shear conditions, and that would represent a Newtonian material, a sample like an oil or a solvent. However, in modern formulations, there's usually multiple components in the formulation, and these give rise to interactions with one another. And as a result, we end up with an idealized flow curve, typically like this, where at low shear conditions we see Newtonian behavior. However, at a certain shear condition, we see a decrease in the resistance, the viscosity, as we overcome the interactions between the various components. And finally, we go into the higher shear conditions, we overcome all these interactions and we enter a second Newtonian or infinite shear plateau. Now, if we consider the low end, this is where it's going to be of interest for things like storage, or sedimentation, or leveling processes. The power law region is going to be interested for predicting draining, pumping, or chewing processes. And at the high shear conditions, we're of course interested in things like mixing, extruding, brush applying, rubbing, or lubrication. One of the other unidirectional techniques is the step transient creep test, where we apply a small constant stress and study the sample's deformation with time. This small stress can be simulating gravity acting on a particle, and so can be used to predict sedimentation, shelf life, or blooming effects in emulsions, or gravity acting on a coating of some form, so we can predict sag or coating behavior. With rotational rheometers rather than viscometers, we have an additional test mode of dynamic testing. This is where the sample is gently probed with small oscillations where we can control the amplitude, the stress or strain. We can monitor how the sample changes with time with a constant amplitude. We can keep a constant amplitude and change the frequency, the time scale, or we can keep a constant amplitude and change temperature in the temperature ramp or temperature sweep. TA Instruments modern rheometers, we have a unique functionality. We can actually do a vertical deformation. This gives us a linear DMA function. So what is oscillation? Well, it's typically a small-scale sinusoidal deformation of the material. The user can define the stress or strain amplitude, the scale of the deformation. They can control the frequency, which represents the time scale of the oscillation process, and they can keep a constant temperature or change the temperature in the background. By controlling these individual parameters, we can learn about our material. So we can see how robust it is by increasing the amplitude. We can change frequency to see the time scales of processes. Simulations of such things as rubbing or applying a cream or lotion. We can look at changing nothing and studying how the structure changes with time. So we can study a gelation, a reaction, processes like drying. And if we change temperature, we can start to simulate cures, or we can look at transitions like TG and melt. The output of any oscillation experiment is the modulus values. G prime is the storage or elastic modulus and relates to the amount of energy stored and can be recovered. This defines how solid-like a material appears. G double prime is the loss or viscous modulus and relates to the amount of energy lost and cannot be recovered, and so how liquid-like a material is. In the graphic on the left-hand side, we see two types of balls. The bouncy ball, the super ball, bounces high because it has a very large G prime and only a small G double prime component, so there's only a small amount of energy lost. However, with a tennis ball, the G double prime component is much larger, and so more energy is lost and dissipated, and as a result the ball doesn't bounce as high. So, if G double prime is very dominant, the sample appears as a liquid. When G prime and G double prime are about the same, then the sample appears like a gel. As G prime becomes more dominant, the sample looks more solid-like in appearance, and if G prime becomes too dominant, the sample not only becomes cuttable, but can become brittle, as there's no G double prime to dissipate any energy. When performing many of the oscillation experiments, we typically try to remain inside what's called the linear viscoelastic region, the LVR, represented here by this plateau of G prime. Beyond these limits, we start to destroy the sample structure, as seen by G prime dropping. Visual aids like these Lissajous plots give us another way of checking linearity because we check for symmetry in the output. Another observation of this amplitude sweep on the hair gel is that we can see there's a clear peak in G double prime, which means that there is a greater energy dissipation as the sample structure started to break down. This is a clear indicator that the sample had some sort of gel-like associated network. Radiological parameters give us a spectrum of behavior. At one extreme, the sample is a liquid, a viscous fluid, and we focus on the viscosity. But as the sample shows more solid-like behavior, our attention switches to the shear modulus of G prime and G double prime, and maybe to the Young's modulus when we deal with the DMAs. A ratio of these moduli can give us a value of tan delta, which is on a scale of infinity to 0. When it's closer to infinity, the material is a liquid. When it's closer to 0, it's more solid-like. So let's look at rheology applications with soft biomaterials. Hydrogels and creams are soft matters that contain high levels of liquids such as water or oil, and have uses in a wide variety of applications including tissue engineering, wound patches, drug delivery, contact lenses, or even superabsorbent materials like nappy linings. Rheology can provide key information on the gel formation to gel strength on different formulations. A natural polymer like hyaluronic acid and natural polysaccharide, which is commonly used in the pharmaceutical, biomedical, and personal care industries, is a typical building block of hydrogels. The rheology can be used to evaluate the viscoelastic properties as a function of the concentration in ionic strength, the molecular weight, a degree of cross-linking, or any other formulation variant. The graphs show that the material, this natural polymer, has a typical response similar to a conventional thermoplastic or polymer melt material. On the left graphic, we see a frequency sweep, where the crossover of G prime and G double prime modulus is an indication of the molecular weight and molecular weight distribution. And that data can be converted by what's called the Cox-Merckx rule to produce the graph on the right, a prediction of the flow properties of the material, which can give us an indication of how easy it is to apply. Hyaluronic acid can form gels, and these can be used as lubricating agents during abdominal surgeries to prevent adhesion, and also for joint lubrication or wound healing. Rheology can be used to monitor the gelation reaction, and in the left-hand side we see the changes in the modulus with time. The sample starts as a liquid with G double prime dominant in green, but at the characteristic gel point, the crossover, the material starts to become elastic dominant. At the end of the gelation, we can then perform a frequency sweep, as shown in the right-hand graph, and here we can see a typical response to show the gel strength of the material, G prime dominating G double prime by a long way, but also being frequency independent, clearly indicating this material is a gel-like structure. With isothermal cures, kinetics can be affected by temperature. In this example, a two-stage cure of bone cement, where it initially starts off as dough-like material to enable the surgeon to fill the hip cavity and insert the pin. The final second stage cure is seen to be onset earlier with an elevated temperature. This is no surprise, as it's common to use temperature to change the kinetics. We heat things up to accelerate the process. We cool things down to slow it down by simply adding or removing energy. It should be remembered that some gelations are initiated by temperature, but in this case this is a thermoreversible gelatin gel. The material becomes a liquid on heating, but on cooling reforms a gel. It's not surprising that gel strength might depend on concentration, as dynamic frequency sweep tests can be used to compare gel strength at the application temperature, in this case 10 degrees, where with increasing amounts of gelatin we can see a firmer gel being formed. At 1% it's actually a liquid, never formed a gel, but at higher increasing percentages we see a gel formation with frequency independent results and higher magnitudes of G prime, the storage modulus. Step transient creep data can be used to give us an insight into the damping characteristics of associated cross-link network structures. When we look at the creep ringing or the oscillation seen in the output, we can judge by the amplitude of the ringing. It's an indication of the strength of those links, those interactions. Whereas the damping of the ringing suggests the interference from neighbours, maybe the number of things connected, the cross-link density. This experimental data for a step transient test shows the creep ringing of Voltarol Imogel and an alternative brand. The blue signal from the Voltarol clearly shows a larger amplitude of ringing, suggesting a weaker network interaction. And the fact that it has a slightly longer dampening also suggests that perhaps there's slightly less connections in the network. Performing a dynamic amplitude sweep, we usually try to define the linear viscoelastic region, a region where we're non-destructive. But beyond that, we start to damage the sample structure, and as a result, we're overcoming the interactions present between the particles or molecules in the material itself. So if we have a narrow distribution, the interactions will be the same, and so it would take a similar force to overcome those interactions. However, with a broad distribution, there'd be a range of interactions, which means a broad range of different forces needed. So the amplitude sweep can give us a clear indication of a narrow or broad distribution. In this example, we see the amplitude sweeps for two materials, Voltarol, Imogel, and its alternative brand. And if we consider that G' is an indicator of the number of interactions, and the length of the LVR the strength of these interactions, we can see that the alternative brand has a much higher G' and longer LVR, being a more robust and narrow distribution. We can also see that there is peaks in G' that indicates that both systems have an associated gel-like structure. Here we see the flow profiles of Nurofen gel, a product typically used for joint aches and pains. On the left-hand side, we see the viscosity profile against stress, or force. And it's clear that the material has a high viscosity at low force, but breaks down readily to a much lower viscosity to make it easier to dispense and to apply. The same data is plotted on the right against shear rate, so we can see how the viscosity changes with the rate of application. Here we see examples of dynamic measurements made on a typical formulation for a lotion. On the left-hand side, we see a frequency sweep, where G', G', and tan delta appear to be frequency independent. And with G' dominated and tan delta close to zero, we see a gel-like behavior suggesting strong repulsive forces, and this would indicate a long-term stability. On the right-hand side, we see a structural recovery measurement done with a dynamic time sweep, where we can monitor how quickly G' and G' recover post-shear application. The faster it recovers, the less likely the sample will flow out after application. Dynamic time sweeps can be used to look at structural recovery, to simulate the stages of application. By performing all three steps in oscillation, we can compare the structure before, during, and after application. So as we start inside the linear viscoelastic region, we look at the sample at rest, undisturbed. If we work outside the linear viscoelastic region, we can damage the sample structure, simulate application, and here it's clear that G' starts to dominate G' by a long way. The sample is more liquid-like and so easy to apply. And finally, if we go back to inside the linear viscoelastic region, we don't destroy any structure as it forms, so we can study the structure recovery. When G' dominates, think of the sample as a solid-like material. It's less likely to move, so it stays where it is. It forms a barrier. If the rate of recovery is slow, then the sample can spread by itself easily, e.g. a lotion may soak into the skin. If we consider the high temperature performance of these lotions, one of which is for UV protection, we can see that at G', for sample A, decreases at 37. As a beauty lotion, it makes sense as this allows the viscosity to drop and becomes easy to distribute and penetrate the active ingredients into the skin. The G' for sample B, with the UV protection, withstands higher temperatures. Again, this makes sense since it's designed to work in higher temperature conditions, like the beach. G' for lotion C, however, shows an increase before dropping, and if we look at tan delta, we see it decreases. This suggests that there's some sort of phase separation for lotion C, and this would be an undesirable product. Tan delta for A and B increase above 37 degrees, and this is probably a reflection of the wax crystals melting, which lowers the modulus. An extension of the unidirectional flow testing is by using the tribogeometry geometries. This modular design with quick and easy methods for interchanging geometries to investigate the coefficient of friction. Tests can be performed in dry or lubricated conditions, and in certain cases the geometry surfaces can be customized, like an N-transport to represent artificial skin. In this example, we see the coefficients of frictions for several lotions. At 37 degrees body temperature, and with a transport substrate to represent the skin, and a 3 Newton load being applied to represent the pressure from the fingertips on applying these lotions, we can look at the sliding speeds to simulate what the coefficient of friction would be on application. In this case, it is clear that the green curve, the top one, has a much higher coefficient of friction and may be seen to be uncomfortable in application processes. Biomedical adhesives can be used in biomedical device assembly, or for hard or soft tissue attachments, such as fillings in dentistry, or for wound closures. Rheology can help to guide the adhesive process in understanding its application properties and its cure properties. Rheological measurements can also correlate to the tack or peel performance of the final product. For adhesives, the tack and peel performance can be seen in a frequency sweep. At low frequencies, representing long timescales, we can predict if a sample will show creep under a constant load. At higher frequencies, shorter timescales, the tack, the application rates, or the peel performance could be assessed. A logical extension of this is the Dahlquist criteria, which specifies a range of G' modulus values where peel would be successful. An additional way of looking at adhesives performance or properties is to look at its temperature dependency. By performing a temperature ramp on this pressure-sensitive adhesive, we can identify several key temperatures or modulus values. The glass transition, the TG, can be identified by a peak in tan delta. One of several modulus crossovers, but at the highest temperature, we finally see the liquid dominance of the modulus G' over G', and as such above this temperature would be a sensible application conditions. The Dahlquist criteria specifies that G' has to be equal to 300,000 and gives us a Dahlquist temperature. And in the midpoint of the two later crossovers in the rubbery region, we can see the plateau modulus value, and this can be correlated to cross-link density. Adhesives performance can also be assessed by axial testing or tack testing. The sample is sandwiched between two parallel plates, which are separated at a constant linear rate. We measure the axial force, the pull of the sample, and resisting this separation. With a cross-link pressure-sensitive adhesive, we often see a rapid build of axial force before we see the sample start to peel off one of the substrate surfaces. With a non-cross-link pressure-sensitive adhesive, we often see a slow build in axial force as a filament is formed. At some point, this filament fails and we get cohesive failure. For dental adhesives, the cure is usually initiated by exposure to UV light. On modern rheometers, we can measure the rheological properties during such exposure. Here we can see G star, the complex modulus, or how stiff the material, change from a low value to high value rapidly. The kinetics of this reaction could have an adverse effect on the handling timescales for the application process for the dentist. With adhesive cures, we often see an associated volumetric change or shrinkage. Shrinkage can cause fractures in the process. In this example, using the rheometer's UV curing test method, we can quantitatively monitor the shrinkage force during the cure, or see the dimensional shrinkage. So on the left we keep a constant gap, we see the force build up between the plates, and on the right we enable the gaps to be changed to follow the volumetric change in the sample by maintaining a constant axial force. So as the pressure builds, the plates are lowered. Drug capsules are typically made of gelatin or polysaccharides such as HPMC. Rheology and DMA can help to investigate the optimal capsule processing conditions and also evaluate the capsule's properties with regards to controlled temperature and humidity. In this example, we're looking at a gelatin capsule and its mechanical strength versus temperature and humidity. By performing a dynamic temperature or humidity ramp experiment, we can help evaluate the mechanical properties, the stability of a gelatin capsule at different storage conditions. In this example, we are ramping the relative humidity at isothermal conditions. It is no surprise to see that the sample's mechanical strength is seen to drop off earlier at higher temperatures by the decrease in G'. The relative humidity accessory is available on our DMA 850 and on the HR range of rheometers. With an HPMC gelation mechanism, a thermoreversible gelation, we can go from intramolecular interactions to intermolecular interactions as we form a cross-link network. On heating the material, we initially see a decrease in both modulus values as expected, but at some point the gelation reaction starts and we see a characteristic gel point or gelation temperature above which gelation will be initiated. Capsule manufacture processing normally involves a hot pin being dipped into a cold solution above this temperature. Polylactic acid, or PLA, and its composite materials are FDA-approved synthetic degradable polymers and show many good characteristic features such as biodegradability, good processability, and low cost. PLA has been widely used in many biomedical applications such as orthopedics, drug carriers, stents, tissue engineering, and antimicrobial agents. How does rheology help? Well, rheology can be used to investigate processing conditions, the temperature and shear, for the polymer processing technologies including extrusion, injection molding, and blow molding. Rheology also provides a quantitative evaluation of any end unit performance of the PLA-based products. We can investigate effects of plasticizers, stabilizers, pigments, or fillers. The PLA melt temperature ramp cycle, we can use a dynamic temperature ramp test on the rheometer to investigate the appropriate extrusion temperatures and also cooling conditions. As we heat up the material, we finally get to a temperature where we see the melt, and therefore the sample will be easy to extrude. On cooling, we see a slow build of G' storage modulus until it becomes dominant, and the sample will now hold its shape. Another way of looking at this is on the right-hand side, where we look at the complex viscosity, the overall stiffness of material. A dynamic temperature ramp can be used to predict the performance, in this case of a PLA end product produced by slow cooling after extrusion. In the example here, we can not only see the mechanical properties of the material, but identify its glass transition temperature by the peak in tan delta. Biopolymers can be used to make catheters and these can be measured in their final form. Torsional clamps can be used to gain the shear modulus values when the sample is twisted, whereas the DMA mode clamps can be used to gain information about the Young's modulus values, either under elongational or bending deformations, depending on the clamps used. It should be remembered that torsional and linear DMA, shear and Young's modulus measurements are only possible on the TA instruments rheometers due to their magnetic bearing and FRD design, which makes it a unique capability of our rotational rheometers. When it comes to the catheters and seeing how flexible they are, the results may depend on the form of deformation applied. In this case, when the sample is stretched with the Young's modulus on the right, we see little or no difference. However, when we take the same samples and twist them, the shear modulus values of G' show an array of results, suggesting some are more flexible than others. In summary, rheology is a powerful tool for investigating viscosity and viscoelastic properties of biopolymer materials and biomedical devices. The materials evaluated by rheological methods can be either liquids, soft solids, gels, or solids. The viscosity can be measured in relation to the shear conditions and can be used to predict dispensing or application performance. The viscoelastic properties can be monitored as a function of time, temperature, frequency, or amplitude. The rheological measurements information can be used for basic research and product development, troubleshooting manufacturing problems, or general QC analysis to distinguish between batch-to-batch variation. So rheology has a role in everything around us.
This series will be talking to many of these material properties, how it can help you measure, and how it can help you understand your material better. Next week's talk will be on applying micropolarimetry to medical device testing, presented by Marlon Susgrip. We hope to see you then. We will now take some time for questions. Whilst we collect the questions that you've been submitting during the talk, we'd appreciate it if you could answer a short poll to help us prioritize how we can help you better. Okay, thank you everyone. I will start the questions that are coming in. Ask Pete. So at the end in the catheter example, you mentioned that the rotational rheometer can also be DMA. How is that possible, and are there any benefits?
Thanks, Ben. Thanks for the question. Very simply, on our rotational rheometers, both the ARES and the HR product range, what we call magnetic thrust bearing. That means that we are levitating the moving parts of the rheometer in a magnetic field. And our clever bods in R&D have decided that the easiest thing you can do is if you actually control the magnetic field, you can make it oscillate in a vertical direction as well. As a result, we can do linear movement rather than just rotational. So the same rheometer can be used rotational-wise with the thrust bearing in a fixed plane of rotation. Or it can be made to oscillate gently up or down on a small scale, so we can measure films, fibers, or stiff bars, bending, or like that. So we've tried to give the ability of using the same instrument in two different ways. Benefit for that, certainly in the higher soft metal materials profile, is that things like hydrogels can be measured in both getting the shear modulus, P primes, but also Young's modulus. And you'll often find that as materials become harder, they start to focus on the Young's modulus data, E prime, E double prime. And those are used to deal with fluids, then to talk E prime and G double prime. Now you can provide an overlap of both systems and temperature controls. So hopefully that's answered that.
Okay. The sound quality isn't the greatest, and you're dipping in and out there. Just so you know, Pete. Okay. We have another question on so for polymer melt and natural polymers, what are the main tests of interest to fully characterize the material?
Okay, so in terms of a concentrated solution or a polymer melt, as we will call it, in terms of materials that have gone beyond the melt transition, you have a concentrated amount of potential interactions within the sample structure, polymer chains themselves. And that interaction is fairly unique and defines the type of product and rheological properties you're going to have. So with the rheometers, we tend to do a number of tests. One main focus is usually to do what we call a frequency sweep, which I mentioned a few times in the presentation, which provides a spectrum, and it's related to the time scales of the molecular actions and interactions. So if you take a frequency sweep of a material, it's a fingerprint. It's unique. So if you take a second material or a slight variation, you will see the differences in the frequency sweep data. That same frequency data can be converted to provide data about how the material will behave at high shear conditions or low shear conditions. So it's one test does quite a few things, and that's something that the polymer industry uses routinely. The other side of it is that, of course, it relates to timescales of processes. So it can be used to look at cyclic movement, brushing, rubbing, or anything else. The other side of it is that in terms of understandings, we talked about dispersion qualities and molecular weight distributions. That comes from that sort of information as well. So just a simple frequency sweep can give you a visual check on how your molecular weight is changing, how the distributions are changing. And that works for concentrated polymer solutions and the polymer melt. And in terms of any other tests we deal with, the other main one we tend to look at is temperature. Transition temperature to understand the TG and the melt conditions. And somewhere during those measurements, there's several types of different regions. I think I mentioned it once in the example, the pressure-sensitive adhesive, that in one region you can actually link back modulus values to things like the cross-link density. You've got a network that's reacting. So quite a few simple tests that can be done on the rheometers routinely, and then of course you can switch on to PMA.
Okay.
That one. Sorry, Ben, I can't hear anything, so I'm just checking the silence means you've done it.
Okay. Yeah, it may take a minute for them to follow up with a follow-up question, because obviously they have to type it. So we'll answer another question in case they're following up on that one. So I've heard of a term called slippage. What type of samples is this likely to be a problem for, and how can I tell it exists?
Okay, well, slippage is usually going to be something you're going to encounter mostly with people dealing with creams, lotions, basically the hydrogel creams or things like that, where you've got an oil-water emulsion or vice versa. The two main reasons for slippage that you encounter during an experimentation in the rheometer, bearing in mind that the rheometer, the idea is to keep one surface of the sample static, the other side to be deformed. You need to get a nice uniform shear through this. If we get slippage, it causes a problem because we effectively lose contact with our samples when we're measuring it. Now, the two forms of slippage arise from either the sample does not like the substrates, materials, the geometry. So things like silicones won't wet the well, some metals and some other materials. So that causes basically a lubricated slip. The other major reason people have problems with slippage is large concentrations of particles or things, which causes what we call wall depletion. Now,
we talked about particles there as an example, where you can imagine that if you've got too many particles jammed into a material, they don't tend to want to be right at the edge. You can't have half a particle. You only have one solid round particle or whatever shape it may be. So at the end, naturally, you can't get so many close to the edge, the top or the bottom, in contact with the geometry. So there's a concentration gradient developed. The issue with when you're dealing with hydrogels and creams and lotions and latexes and emulsions, stuff like that, is you're going to have issues where the emulsion droplets can't exist in half of an emulsion droplet. It's a full droplet. So you get the same concentration gradient. And what you often find is you'll get a very thin layer forming between the surfaces. And again, that just acts like an aquaplaning zone so that your geometry will slide on the top lubricating layer rather than grip your sample. So the very simple solution we deal with that is that we literally roughen the plates like you do on your car tyre, so we can grip the sample and take out the troublesome layer, so that we're really measuring the bulk properties of the material rather than just letting the geometry slide on the surface. And this can also happen if you do measurements with temperature. If you, for example, take a sample in there and it heats on one side but is still solid on another, it's a bit like butter in a frying pan, it's quite round. So we could have roughened plates on the top, or we could have bottom, or on both, solve the issue. The indications in terms of results is when you're trying to do some of your testing. If you're doing a flow test, you'll get a sort of step in the data where it slips. Just like you can imagine watching somebody walking along and slipping on a banana skin, they slide suddenly along distance. We'd see that in a sort of two-stage step in our flow curve. And if it's happening during an oscillation experiment, we normally display what's called waveform. I think I mentioned a couple of times in the presentation, where we're looking at the sinusoidal movement backwards and forwards. And if you have slippage, you don't get sine waves, you get really like sine waves that don't fit in the screen. There's a flat bottom, like tabletop mounted. And what you've got there is that the sample, as it tries to do the deformation, slides to the end and then picks the oscillation on the way back down again. So we've got visual aids that can help us pick out this. You can do other things like vary the gap, etc. If it's dependent on that, to eliminate. But the general thing to point out is for most people here is if they are dealing with emulsions, oil and water emulsions, vice versa, then they're prone to having this. And of course, they can see it in real life because when they open up their product after storage for a couple of days or whatever, if they can see a thin layer of liquid forming on the top, that would lead to slippage on the experiment. So it's visually, you can assess before you get it on the reometer. There's a solution when it's on the reometer by using roughened plate, or often called crosshatch, braided or sandblasted braided material.
Okay, thank you. Just to mention at this point as well, we've got a few people asking about the slides. So after this event, we will be sending a follow-up email with a link to the recordings, and we'll also try and add some of the FAQ for those of you that are struggling to fully understand the sound quality. So, but while it's going on, there's another question. It's another in-depth question for you. So how to evaluate the disentangle and entangle degree of polymer?
Okay, I think in the time we've got, we're going to be struggling to fill all that information in, especially if you can't hear me very well. But very simply, it does become more complicated in terms of understanding that when you're measuring rheology, you're generally looking at what we call bulk properties. So when I'm measuring on a rheometer or anything, I get a response, which could be an average of a number of interactions and behavior and orientations within the material. When it comes to disentangled and entangled degrees of polymers, the classic scenario of that is to work out where we're looking for in terms of understandings, because the rheometer will see a difference, but it might not be able to assign certain amounts on disentanglement or entangled without doing additional testing. Some of the examples, for example, if it's disentangled, then that means the polymer materials will be able to move separately, independent of each other. Now, one discussion we have with things is whether they loop around another so they become entangled, and in which case, when you try to pull one, you're going to pull another because it's physically hooked around it. And as a result of that, you can start to look at larger scale deformations and things like that to understand if materials are either cross-linked or physically entangled. Now, there are more complicated ways of looking at this, but keeping it simple, that's how we approach it. We're going to be doing deformations on not only a small but on a larger scale to see if effectively you pull one loose thread out of, say, an example of a shirt or a cardigan or a jumper or whatever, it just pulls out and that's it, you've lost it. That would be a disentangled system. Something that's entangled, it's like you pull it and it snags, it pulls the rest of the shirt or the jumper with it, like a knot in a string. So as a result of that, you can start to learn that there's some quality there to investigate further. And we do things like extensional testing on the rheometers, certainly for polymer melts, where we can actually do extensional tests to large Henke strains to actually see if there's what we call a strain hardening effect. We also see that extensional type behavior if you do an amplitude sweep and see the modulus is increasing, because that suggests that the thing is actually trying to deform and it's resisting more. So there's a number of things, but there are people who are cleverer than me who will start to fit equations and predictions and start talking about repetition and all these things, which, again, some of the information you can get out of the frequency sweep profiles. It depends on the time scale of the type of things that are being involved with. So there is sort of quite a few of our customers are trying to really get clarity on their samples by looking at these properties. But the challenge is it's not just a simple test. There can be a multiple of tests you have to do to be able to pick up the information or discredit, or basically discern between samples that are just showing their properties because that's what they are, or whether there is a subtle difference, because we have to identify the bulk properties and look on a more macroscopic level for that. And that's why there's an outbreak of materials that are looking into just mathematical treatment to understand these things so that they can perhaps identify subtle changes. But yeah, I would say that it's not as easy as we would like it to be. We can certainly define cross-linking and stuff like that if you know the chemistry exists, but if it's just physically polymers entangling with each other, then it's another interaction. And defining the interactions, the best way, perhaps like most scientists will do, is we look to see if we can manipulate the materials we test, give us an example of, well, we know that this shouldn't really be entangled and this should have some entanglement, and measure that on the rheometer to see if we can see where it's affecting the data. And then, of course, correlate it with other devices or other techniques, whether it's optical or anything else. And for that reason, that's why there's been an outbreak of things like optical tweezers techniques, where they literally grab one end of the polymer and pull it and see if it comes out like a game that you play with a kid, just pulling out a straw and seeing if the ball falls or whether it snags and catches and it's then entangled. So I hope I've made that clear, because I know that some of you won't be speaking English and the sound quality is not so good. But yes, you can get some indications, but it won't give you percentages so readily without some extra work.
Thank you, Pete. So yeah, hopefully that answered that question. So what we can do is, I say, we will send a link to everyone with the recording. But people that have asked specific questions in the chat, we will contact. We can contact you directly with a written answer as well. Yeah, so I think with that, thank you everyone for attending. This is part of a series, so there'll be another one this time next week on microcalorimetry. So thank you to Pete for the presentation. I hope everyone found it very informative, and we'll speak to you again hopefully next week. Thank you very much, and goodbye.